right, nerds, let's try doing this meiosis puzzle together. So the instructions say, study the differences in the cells, then move the cells around to put them in the correct order for meiosis. Wait a minute, KJ, you didn't actually tell us the correct order for meiosis. Right, you guys have to kind of figure it out. We can solve it together. It says, finally add the appropriate labels next to the cells. Okay, so let's go ahead and just glance at the cells here really quickly and see what kinds of things we notice. So for example, I see 2 chromosomes and they're lined up in the middle of the cell. I see 2 chromosomes lined up in the middle of the cell over there also. I see that here's a cell that's dividing and it's got bivalent chromosomes. It's got 2 sides to the chromosomes. This, I see the cell is dividing, but those aren't bivalent chromosomes. Those look more like chromatin, like it's all unwound, it's messy hair. Oh, wait a minute. I recognize this cell. That cell looks like interphase, right? That's just the normal cell. Why don't we try putting that one up at the top? Let's move that cell. Let me squeeze out a little bit here. There we go. I'm trying to magnify it for you. All right, so let's put this cell all the way up at the top. I wonder if it works better. Ooh, my touch screen works really nice, so I think I'm going to put it up at the top. Okay, so I'm pretty sure that that's interphase. So let's go take a look at the labels. Is there one that says something about interphase? There it is, interphase. This one right here. So I'm going to grab it and I'm going to put the label right next to it. So it says that the cell here is diploid. Oh, right. It starts diploid and then it becomes haploid later on. All right, so let's try some other cells. So I don't know, this is pretty confusing. I'm not sure how to do this. But I do see some cells that I kind of recognize, so I'm going to see what I can figure out. So these cells that are dividing, I remember that KJ said the cell divides 2 times. So I see one cell that's definitely dividing. I'm going to put that about here maybe. And then I see 2 sets of cells that look the same, this one and this one. I'm using my finger to move it. That's easier than using my trackpad. Those 2 look the same, so maybe those are the cells that are at the very end. Maybe those are the cells at the end that are going through telophase. And it says here, it reminds us that the stages of meiosis are called IPMAT, PMAT. Oops, I'm trying to highlight just that. There we go. IPMAT, PMAT. So interphase, prophase, metaphase, anaphase, telophase. So telophase is when it splits into 2 cells. So let's see if we've got some telophase labels. We've got telophase with a 1 by it, and then we've got telophase with a 2 by it. So I'm going to guess that this first one is telophase 1. So I'm going to grab this label and put it right there, telophase 1. It says the 2 new cells have half the DNA, so now these cells are haploid. Okay, well, if that one's telophase 1, then this one up here must go down for these cells down here. Maybe I'm even going to put the label in between them. Hold on. There, let's see if we can put the label kind of evenly between these cells. Trying to move it around a little bit. There we go. Okay, so we've got our telophases placed. Now we need to do prophase, metaphase, anaphase for each of them. I think prophase is hard to recognize, but metaphase, what were we supposed to remember with metaphase? Metaphase middle. That seems like that's fairly easy to solve. So I see this one's in the middle, and I see this one's in the middle, and I see this one there in the middle. Now, which one would come first in the first half of meiosis? Well, it says here in telophase that the 2 new cells have half the DNA. So if I look at this cell and this cell, they both only have 2 chromosomes in it. But this cell has 4 chromosomes. So I'm going to guess that this one goes up here, that this is metaphase at the beginning, and then the chromosomes were separated from each other into the other cells. So I'm going to say maybe these go down here across from each other. So this cell on the left became this cell here, and this cell on the right became this cell over here. Okay, well, I'm going to go with that and see what we can do. So those are my metaphases. Actually, and I bet I could add my labels. So here's the label metaphase 2. I could put that right between the ones that are in the middle. And then metaphase 1, there it is, metaphase 1 lined up with homologous partners. Oh, that makes sense. Look at them. They're side by side with their partners. Okay, that works. Okay, now we need to do anaphase. I think prophase is the hardest one, so I'm going to go to anaphase. And I remember the word we remember with anaphase is anaphase apart. And here it says anaphase 1, the homologous partners are separated. And then where's my other anaphase? This one just says anaphase 2. It doesn't give me any special details. Let's see if I can see ones where they're being pulled apart. Okay, this one's being pulled apart. This one's being pulled apart, and this one's being pulled apart. And you know what? They kind of look the same, but if I look at them really close, I see that these ones are still bivalent. They're still with their partners. They're still with the second, so they have two sides on them. Whereas these ones, they're monovalent. They have been ripped apart from the bivalent chromosome. So I'm going to guess that these two are the ones that are at the end, and this is the one that's near the top. So let me pinch it closed again. Let's see if I can grab this one. There we go. Anaphase 1. And that's the homologous partners are being separated, but they're still bivalent. And then in anaphase 2, the chromosomes, the bivalent chromosomes are being ripped apart. That makes sense. All right, so all we have left now are the prophases. So we've got this one, this cell, we've got this cell, and we've got this cell down here. What I notice again, if I look at them really closely, I see that this cell still has 4 chromosomes in it, but this one only has 2, and this one only has 2. So the ones that have 2, they've already gone through cell division. They are haploid. They have half the DNA. So I'm going to guess that this one is the one that goes up at the top. Oh, and you know what else? I see that they're crossing over. They're trading their pieces of DNA. And I think that's part of what's going on with prophase 1. So I need to move these around a little bit to give myself enough space. I'm going to cough, so excuse me if I start having a coughing fit. Oh yeah, there's prophase 1, there's the label. And crossing over occurs there. And then these two must be the prophases that go here. So when this cell splits, there's now 2 new cells that are in prophase, and that's this guy, this guy, and this guy. So I'm going to call that prophase 2. I think we're all done, folks. We've got an interphase, prophase 1, metaphase 1, anaphase 1, telophase 1, and then prophase 2, metaphase 2, anaphase 2, telophase 2. That's it. When you get it all set up, you can go ahead and submit it.